Greetings, I'm on the east side of the large island of Hawaii, and I thought this was a beautiful place to talk about protein catabolism, and you'll know why in just a moment. Recall that much of our coverage has actually alluded to the way in which amino acids can either be built or can be broken down, and the way in which their carbon skeletons can enter into metabolism depending upon the needs of the cell. So there are a couple of ways in which we can think about amino acids as being utilized. They can be utilized for energy. They can be broken down fully into CO2 and water, all used up, worn out. But also we recognize that depending upon the needs of a cell, they may be utilized to build glucose. So recall that the skeletons of amino acids could be used as gluconeogenic precursors, serving, for example, the glucosalanine cycle, right? And be used to build sugars. Now, if someone is in a truly starving state, we also know that they could be used as the precursors to synthesize ketone bodies. So amino acids can be broken down for that purpose. But of course, if you've just taken in a lot of Hawaiian pig and you just have excess protein, it could also be used in the synthesis of fatty acids. So we could see these proteins getting used in a variety of ways, but no matter the way they're utilized, they're still going to have to be detoxified. Remember that amino acids, proteins in general, are toxic. So these are going to have to be deaminated. So they lose that amino group, and it, of course, forms ammonia. Now, you can't really envision wanting to drink your household ammonia cleaner. That's pretty nasty, pretty damn toxic. That's true of proteins too. So ammonia has to somehow be cleared from the body. It's not something you want to hang around. And that's going to be the job of our liver. Now, depending upon the type of organism, the way in which we secrete toxic ammonia waste is going to vary. And that's why we're here, because the aquatic organisms, I was actually just looking at a little crab. Aquatic organisms are going to be able to secrete a much more concentrated form of ammonia because they go right into the water, and it's not a problem, right? Because it's diluted. The more aqueous the environment that an organism lives in, the more highly concentrated the toxic ammonia can be secreted. But in us, of course, we're land animals. We're going to have to convert that toxic ammonia into urea, right? Urine. We're going to have to do that, and you can imagine that takes a little more energy than just secreting toxic ammonia. Now, if you think about, say, like desert lizards, they're going to have to secrete something even less toxic. The drier the environment, the less toxic the ammonia form has to be. So they actually convert it into uric acid in order to secrete something really non-toxic. So these organisms that live in the ocean, they can secrete it almost directly, all diffused out and diluted. But us, we have to do the urea cycle. So let's zoom in and take a moment to look at the nuances and details of the urea cycle occurring in my liver right now. This is a big picture view of the urea cycle, and we're going to zoom in on little portions of it as we go. But for now, I want you to notice a couple of things. Notice first that part of the urea cycle takes place within the mitochondrial matrix. Another part of that cycle takes place within the cytosol. So we are actually looking at a switch in compartmentation throughout the cycle. We can also notice throughout the cycle that there are a lot of intermediates towards the synthesis of urea, and one of the things that one of our goals is going to be to watch where the nitrogens that get into that urea molecule come from, and also where the carbon atom comes from. Great. So we're going to see that as we go. We're also going to know as we go that this process, this pathway, is going to be endergonic. It's biosynthetic, it's anabolic, it's synthesizing something. Okay, let's zoom in on the first step of the pathway, beginning by looking at the fact that glutamate is oxidatively deaminated, so it loses its amino group, forming ammonia, and it's an oxidation as well, so it also forms reducing power, either in the form of NADPH or NADH. Let's zoom in on just this region of this cycle. Nice. So what we can see then here is that ammonia, toxic ammonia, is formed from that deamination event that takes place right here. And this ammonia is going to react with bicarbonate. And this is going to form the first intermediate of the urea cycle called carbamoyl phosphate. Carbamoyl phosphate is a very famous molecule because not only is it an intermediate towards the synthesis of urea, but it is also a starting molecule towards the synthesis of certain pyrimidines, so nucleotide biosynthesis, important there. It also, in organisms that don't have the urea cycle, it also is able to be used as a precursor to synthesize arginine, and you'll notice that arginine is the last intermediate towards the synthesis of urea, so if an organism doesn't make urea, it might use this molecule as a precursor to make arginine. Cool. So this molecule very important in numerous ways. And in fact, representative of that importance is that this enzyme right here that catalyzes this reaction, we're not going to look in depth at it, but I want to mention it, carbamoyl phosphate synthetase 1. It is one of the most abundant enzymes in the mitochondrial matrix of the liver cell. It actually accounts for up to 20% of the protein content in the mitochondrial matrix, so it is a very, very, very prevalent enzyme. So we might write down then the ammonia released when glutamate is oxidatively deaminated reacts with bicarbonate to form carbamoyl phosphate. Step 1. Now look at something here. Notice that this is a highly energy consuming step, highly endergonic. It requires 2 ATPs to power the synthesis of carbamoyl phosphate. All right, let's look now at how carbamoyl phosphate is going to react with a somewhat unusual amino acid to form the next intermediate in the pathway. Here we have it, carbamoyl phosphate reacting with this unusual amino acid, ornithine. This is going to make another unusual amino acid called citrulline. So we see that being the next step of the process. Now notice that there is a transporter. In fact, this is a citrulline-ornithine exchanger and allows for citrulline to go out, ornithine to come in. It's interesting to note, actually, that ornithine, when it comes to the urea cycle, is really kind of a catalyst because it allows for the formation of the citrulline intermediate, and then at the end of the cycle, it itself is regenerated and comes back in and does the same thing again. So, if you will, it's regenerated but not consumed, just like a catalyst would be. So we're going to go on to the next step. One thing I want to note before we leave this too far behind is notice that one of the nitrogens that's going to be converted to and be found, if you will, in urea is originating from the ammonia molecule off of glutamate. So that's the origins of one of those nitrogens. Now, citrulline is going to swoosh out into the cytosol, and in the next step, we'll look at the fact that citrulline reacts with aspartate, most often that originated in the mitochondrial matrix and was exchanged for glutamate in the glutamate aspartate translocase. So aspartate and citrulline react with one another in another endergonic, very energy-consuming reaction that consumes an ATP. But wait a minute, it's more like consuming what? Notice it goes to AMP plus pyrophosphate, which this pyrophosphate likely goes to 2 inorganic phosphates. It's more like consuming 2 ATP to power the formation of this next monster of a sexy molecule called argininosuccinate. Argininosuccinate is going to be converted in the next step to arginine. So we see how arginine becomes an intermediate in this pathway. Notice though that the next toxic nitrogen originates from aspartate. So one of them came from glutamate, ultimately from the ammonia there, and one of them came from aspartate. And we have now accounted for both of the nitrogens that will be found in urea that is excreted. Okay, so in the next step then, we see that fumarate is lost,
arginine is formed, and in organisms that do the urea cycle, then this will be lost, this molecule or this moiety will be lost as urea. So we kind of come to the end of our cycle. So argininosuccinate is cleaved, forming arginine and fumarate. So the urea molecule, the carbon came from the bicarbonate in the very beginning. One of the nitrogens came from ammonia. The other nitrogen originated from aspartate, and we finally have a toxic urea that can now be excreted from the kidney. It's transported in the blood from the liver to the kidney and then excreted from the kidneys. And there you have it, the best way for us to get rid of toxic nitrogen. Now, before we leave this, let's make sure that we go ahead and keep track of all that's happened and write down all that's happened in terms of energy and whatnot. So the guanidinium group of arginine is cleaved, forming urea and ornithine. We already mentioned that. And now we'll go ahead and mention what our energy input was like. 4 ATP total, right? Notice here we had the 2 ATP input. Here we had an ATP, but it was hydrolyzed to AMP and PPI. So that's more like the equivalent of 2 phosphoanhydride bonds, 2 high energy bonds. So it's more like the equivalent of 4 ATP that are consumed here. 2 nitrogen atoms of the urea come from the ammonia right here and from the aspartate, and eventually here we have both of those. The carbon that is on the urea molecule came from the bicarbonate that reacts with the ammonia here. All right, that is the urea cycle in a nutshell, and now we have a better idea of what happens to that ammonia that is produced when protein is deaminated. Phew, I'm glad we're done with that. All that talking of urea cycle has made me need to use the little girl's room.